Certain human genetic disorders can come from chromosomal changes such as nondisjunction. So, besides passing on mutations through genes, we can also have actual problems with the chromosomes. So we talk about mutations, but we can have structural abnormalities that actually occur within the chromosomes where a whole piece of the chromosome gets deleted. Or part of the chromosome can actually be duplicated. You can also flip-flop parts of the chromosome, and that would be an inversion. And a translocation is where one part breaks off from one and gets put onto another. So again, these are major issues, and they can get, again, this kind of swapping here with this translocation. It almost looks like crossing over, but again, you would not cross over on chromosomes that are not the same chromosomes. So again, all of these are going to cause major problems. We also have numerical abnormalities that leads to non-disjunction. Again, if we look at this karyotype, there is an X missing here. So if we are missing an X or a Y, we have Turner syndrome. This one has an extra X, and this would cause Klinefelter syndrome. So sometimes we have monosomy or trisomy where we have too many or not enough chromosomes, and that also causes some major issues. When we start thinking about non-disjunction, it's the failure of chromosomes to separate during metaphase. And again, we have checkpoints built in to try and ensure that they're going to separate properly. If meiosis happens, or if non-disjunction happens during meiosis 1, we don't split the homologous chromosomes. And therefore, these have too many. Again, they should only have one in each, and these are missing that chromosome. So when this egg is fertilized with the paternal chromosome, this will result in monosomy, and this will result in trisomy. So if non-disjunction happens during meiosis 2, again, the sister chromatids are splitting apart during meiosis 2. So in this side, they did normally separate, and the homologous chromosome separated, and on this side, the sister chromatids separated, and this is normal. You end up back at diploid after fertilization. However, on this side, the sister chromatids fail to separate, and both of these get pulled into this cell, which causes trisomy in this one and monosomy in this one. So again, all of this still, even the non-disjunction where we have too many or not enough chromosomes, comes back to the genes. So this little extra 21, again, it's almost towards these sex chromosomes, and we get smaller as the higher number get. For trisomy 21, which everyone understands is Down syndrome, that extra chromosome there is ultimately, the scientists have figured out that it's this extra GART gene that builds up too much purine in their blood and causes most of the problems that are associated with Down syndrome. So again, it's like too much expression of a particular gene when you have too many chromosomes. Certain human genetic disorders can come from chromosomal changes such as non-disjunction.